everyone. My name is Nikoleta Vavouraki. I'm a fourth-year PhD student in the Department of Pharmacy in the University of Reading. During my PhD, I'm studying neurodegenerative diseases through systems biology approaches. I have always been interested in studying neurodegenerative diseases like Alzheimer's disease and Parkinson's disease because they can alter our nervous system and thus not only our abilities to, for example, move, but also our perception of ourselves and the world. I'm really hoping that more research in this field will lead to finding treatments for all these people that are affected. So today, we're going to talk about a part of my PhD project in which I studied hereditary spastic paraplegias, a group of neurodegenerative diseases, using networks of proteins. We will talk about both the disease and the approach. Let's start first with the approach. There are multiple ways to try to understand the mechanism of a disease. One common approach is to focus on one gene, which is a piece of genetic information, that when altered, leads to the development of the disease. We can start by finding out what the gene normally does in a healthy person. Let's say that one thing that this gene does is to produce a protein that is essential for maintaining the membrane of our cells in a healthy state. Maybe the mutated protein, let's call it A, can no longer activate the next protein in the pathway, let's call it protein B, and thus it leads to a more rigid cell membrane. When we know that information, we can then design a drug that activates B, bypassing A, to fix the problem. However, a single mutation in a gene can sometimes cause different diseases in different people. And at the same time, a single disease can be caused by different mutations in different genes. So another way to try to find the disease mechanism is by a more zoomed out approach, by studying altogether the genes that when changed lead to a certain disease. Since they can all lead to the same disease, they must have something in common. Maybe, for example, they regulate the same cellular pathway. In order to find these missing connections between the genes that lead to the same disease, one thing we can do is to focus on the proteins produced by each of these genes and build networks based on their protein-protein interactions. Let's start with the basics. Why focus on protein-protein interactions? As we said before, one of the ways proteins function is that they form pathways. In these pathways, proteins pass the signal from one to the next by interacting with each other. So if we collect data from protein-protein interactions, we might find that some of the proteins that when mutated lead to a disease are connected, either through direct interactions or, this is more common, by interacting with the same proteins. If we visualize these connections, we'll see that they can form a network of linked proteins. Such networks are called protein-protein interaction networks. Then we can analyze that network. We can focus on the proteins that lead to the disease and their common interactors. We might find that most of them are part of the same pathway, so that could suggest that the dysfunction of that pathway could be the cause of the disease. Information about protein-protein interactions is produced daily by researchers around the world. This information is then added in databases, through which they can be easily accessed by, for example, querying based on the name of the protein of interest. For my work, I need to access protein-protein interaction data from multiple databases that might have some overlaps and they may present their data in a slightly different format. So I use the online tool PINOT, which was developed by our lab. Let's hear more on why it was developed and how it works from Dr. James Tompkins.
Hi, I'm James, and I was part of the team that developed PINOT, or the Protein Interaction Network online tool, whilst at the University of Reading. PINOT was developed to facilitate access and analysis of protein-protein interaction data from a range of database sources. It enables researchers to obtain a wealth of data derived from years of experimental work from labs all around the world. The user simply queries a list of proteins, and in just a few minutes, PINOT collates, processes, and provides the user with an extensive and quality filtered dataset of reported protein-protein interactions in an easy-to-interpret format for downstream applications such as network analysis. Our motivation for developing PINOT was centred on harnessing readily available data for addressing biological questions relating to the molecular landscape governing how our cells function and dysfunction. Since we're in the process of utilising protein interaction data within several of our current research projects and building a pipeline to overcome data integration challenges, we decided to fully automate the pipeline, and with support from Professor Liam McGuffin here at the University of Reading, we now provide PINOT as a freely available web resource for the research community. Since its development and release, PINOT has been incorporated into numerous research projects, including Alzheimer's Research UK funded work here at the University of Reading. We're grateful that this resource continues to be used and hope it's of benefit for many more research projects in the future.
Now that we saw why we use the protein-protein interaction network approach and why we collected the data using PINOT, let's go back to the aim of the project, the study of the hereditary spastic paraplegias, or else HSPs. The HSPs are a group of neurodegenerative diseases characterized by spasticity and weakness in the lower body. They are complex genetically, as they can be caused by mutations in over 70 genes. Even though they have been described at least 140 years ago, the molecular mechanisms responsible for disease are still unclear. So the first step in studying the HSPs was to get the list of genes that they can lead to it, and collect the interactions of the proteins they produce. These proteins we will be calling seeds from now on, because we use them to build or grow a network. This was done using PINOT. When we visualize the interactions using a software named Cytoscape, we can see that they formed a network. Interestingly, even though some seeds were separated, most of the seeds were linked through common interactors. As we said before, we want to find what links the seeds to each other. So, we filtered out interactors that were unique to one protein and kept the interactors that were common to at least 2 seeds. And this gave us the core HSP network. We can then analyze the core network to find processes that are more common in our group of proteins than what we would expect. We do that by comparing the frequency of a specific process in our proteins to its frequency in all human proteins. If there are any processes that are more common than expected, they're called enriched. This is a statistical process called enrichment. Indeed, there were multiple biological processes that were enriched, as you can see here. To find out which might be more important, we can focus on those with the highest enrichment. A lot of them were related to transport, and more specifically, to transporting neurons. In addition, we performed enrichment of pathways, and we found that pathways related to trafficking were enriched in our core network, which aligns with our previous findings. So we saw that we started from all the known genes that can cause hereditary spastic paraplegias. We made a protein-protein interaction network, and by analyzing it through enrichment, we were able to highlight some potential mechanisms of HSPs. We also analyzed the HSPs using some clinical data, but this is a short video, so you can find more details in a paper. I hope that now you can see how useful it is to create and analyze protein-protein interaction networks in order to understand complex human diseases, such as the hereditary spastic paraplegias. This approach can also be applied to other diseases, such as Alzheimer's disease and Parkinson's disease, because there are a lot of available data in multiple databases in the level of genes, transcripts, and proteins, all of which can be insightful. My personal goal is to continue using systems biology approaches like protein-protein interaction network analysis and mathematical modeling to understand neurodegenerative diseases, because I believe that they might bring us a step closer to our goal, to find a treatment for these people. I would like to thank the Alzheimer's UK Thames Valley Network for honouring me with their early career Research Video Award 2021, and giving me this opportunity to make this video and share my research with you. Also, I'd like to thank my supervisors, without which none of this would have been possible: Dr. Patrick Lewis, Dr. Claudia Manzoni, and Dr. Marcus Tyndall, and also the members of their labs for their valuable feedback over the years. If you're interested in this type of research and you want to chat about it, or if you want to collaborate, feel free to message me LinkedIn. Thank you for watching.